Hello everyone and welcome to today's webcast, GMP Analytical Testing and Gene Therapy Products. I'm Tim Wright, editor of Contract Pharma, and our webcast today is being sponsored by Frontage Laboratories. They're a CRO that provides integrated, science-driven product development services throughout the drug discovery and development process, enabling pharma and biotech companies to achieve their development goals. Joining us today is Jessica Zhang, the Director of Analytical Services and CMC at Frontage Labs. Jessica has been working in biologics testing, especially in cell and gene therapy product testing, for more than 15 years. She has broad knowledge and experience in gene therapy product testing, especially in assay development, qualification and validation of cell-based potency assays, and other cell and molecular biology analytical assays. After Jessica completed her postdoc research at the Department of Pharmacology at the University of Pennsylvania, she had been a senior quality control analyst in clinical vector core at the Children's Hospital of Philadelphia and played an important role in the CMC quality control and stability study of the first gene therapy product, Luxturna, approved by the FDA that targets inherited disease caused by RPE65 mutation. As I mentioned, Jessica is currently Director of Analytical Services and CMC at Frontage Laboratories. And in this webinar, she's going to focus on GMP analytical testing in gene therapy products, introducing 3 critical test methods: AUC, infectivity, and RCL assays. New released FDA draft guidance for industry and human gene therapy products will also be discussed. We will be accepting questions throughout the talk. You can send those in using the box at the bottom of your screen. Questions will be answered after the presentation in a brief Q&A. If you're experiencing any technical difficulties, you can type those into the box as well, and someone on the back end here will help you out. At this point, I will turn it over to Jessica.
Thank you, Tim, for the introduction. Hello, everyone. I'm Jessica Xinhua Zhang, the Director of CMC Analytical Service in Frontage. Thank you for participating in this webinar, as I know how valuable your time is. It is a pleasure for me to present to you all on the GMP analytical testing for cell and gene therapy today. As we all know, cell and gene therapy has grown tremendously in the last 15 to 20 years. The slide shows that the global pharmaceutical market is expected to reach $1.5 billion by 2023, and we can see the BLA and NME approved by FDA in the figure, and the light blue shows the NME approval, and the dark blue on top shows the BLA approval by FDA. Starting 2005, we can see the BLA approval by FDA is growing significantly every year. As you can tell, the previous slide didn't include the data from CBER, and here we can see the BLA approved by FDA CBER in the last two and a half years. So here the dark blue shows the NME approval, and the light blue on top shows the BLA approval. We can see the ratio between BLA and the NMEs is growing significantly in the last 2.5 years. So basically, FDA approved BLA is increasing every single year since 2005. And at the bottom right, we can see FDA approved CAR T-cell therapeutic drugs in the last 5 years. So far, there are 16 approved gene therapy drugs in the market today, with 260 drugs in development. By 2025, FDA predicts it will be approving between 10 to 30 cell gene therapy drugs per year. It also predicts that currently 628 active individual US-based clinical trials for durable gene and cellular therapy comprising 211 indications, 335 preclinical studies, 91 phase 1 studies, 174 phase 2 studies, and 19 phase 3 studies. So by 2030, about 50,000 patients per year may be treatable with gene and cell therapy products. First, let's see what kind of cells can be used for cell therapy and what kind of analytical tests for cell therapy. The cell types that are used for cell therapy include chimeric antigen receptor T lymphocytes, which also called CAR T cells, and include stem cells like human embryonic stem cells, neural stem cells, mesenchymal stem cells, hematopoietic stem cells. Some investigators also invested in induced pluripotent stem cells, iPSC, and tumor-infiltrated lymphocytes, TILs, and dendritic cell and neural killer cells, which is called NK cells. The analytical tests for cell therapy include the characterization of the cell type, cell-based potency assay, transduction efficiency, cell count and viability determination, flow cytometry, and tumorigenicity and immunogenicity. Of course, it will include the safety test, which includes endotoxin and gram staining and the sterility test. Gene therapy is to edit or repair human genes by either AAV or lentiviral vectors. So AAV vector can deliver the gene by direct injection, and the gene expression by AAV vector is a long-term expression. And the lentiviral vector transduces the gene into T cells ex vivo, and then the transduced T cell can be expanded ex vivo, and then the T cell can be infused into the patient. And the gene expression by lentiviral vector is short term, so it has to be infused couple times. So, and also at the same time, the target gene capacity is different for AAV and lenti. So the capacity for AAV is around 4 kb, and for lenti it's about 8.5 kb. AAV vector represents adeno-associated virus. It's often chosen as the vector for gene therapy due to their low immunogenicity and the ability to promote long-term gene expression, and the capsid-dependent tissue specificity. As we can see here, different serotypes of the AAV can be used in different tissue and different organs. So right now, AAV vector representing around 37% of the current advanced therapy market. So as we can see, there's an ongoing demand for robust quality lot release program and also for supporting the large patient cohort involved in clinical trials. The AAV viral vector production includes the upstream and downstream and the final fill as shown in the workflow here. So first, the cell was thawed and the virus seed cell was prepared. And after the cell expansion, the virus was transfected into the cells. And after the viral vector transfection, the cells was expanded, and then the viral vector were collected for purification. After the purification, the viral vector formulation is prepared, and then the final fill will be performed for the viral production. GMP testing for AAV gene therapy products include the identity, the potency, the purity, and safety and stability program. So for each quality attribute, there are different techniques that can be utilized to fulfill the purpose. Like SDS page, the MS Western blot can be used to identify the vector. Real-time PCR, digital PCR, and optical density and the nanosite HPLC can be used for the potency. At the same time, the cell-based potency assay and TCID50 infectivity assay can be utilized to determine the potency as well. And we can see the MS chromatography and whole cell DNA, whole cell protein, and the AEOC can be used to determine the purity of the vector, and then endotoxin Mycoplasma replication competent virus testing can be utilized to determine the safety of the vector. And stability usually include pH osmolar aggregation formation and cell-based potency sometimes for stability program. Frontage have all the analytical equipment and can provide the method for gene therapy product testing, including the titer by Q-PCR and D-PCR and infectious titer, and the empty-full capsid ratio tested by CIEF and CE-SDS, and the impurity assay by HPLC and UPLC, whole cell protein, whole cell DNA, and the residual cesium, residual Benzonase, residual BSA for purity assay. And at the same time, we also provide potency assay which can be detected by cell-based assay, CIEF, CE-SDS, HPLC, MS. Meanwhile, we also provide the testing on sterility, BioBurden, Endotoxin, Mycoplasma as well. We provide analytical testing on AAV gene therapy products, include the titer, the genomic titer and infectivity assay, the purity by SDS page, CE-SDS, silver staining, plasmid integrity by restriction enzyme digestion and gel analysis before package. We also help our client to detect the gene expression after AAV infection. It can be done by ELISA, by Western blot, or bioactivity assay. For capsid identity by ELISA, residual plasmid by Q-PCR, cell genomic DNA by Q-PCR. As mentioned, the sterility test for AAV vector can be performed by BioBurden sterility, gram staining, etc. We also perform Mycoplasma by Q-PCR and Endotoxin by chromogenic assay. In addition to that, hot cell protein, residual Benzonase, residual BSA, empty capsid to full ratio by CIEF and CSI. In addition to the lot release, we also perform long-term stability study for AAV gene therapy products, which also include color, appearance, pH, osmol, and cell-based potency assay as well. PCR-based titer assay is very important for both AAV gene therapy release and the stability program. We perform real-time PCR and the digital PCR. The digital PCR provides advantages to Q-PCR due to its extreme sensitivity and ability to detect real events. And similar to Q-PCR, it relies on techniques like probes and primers, but it identifies target DNA sequences in amplified or droplets. So the amount can be calculated based on a simple Poisson distribution. The experiment can be scaled up for even more sensitivity. So that's how we perform the digital PCR for AAV vectors. Next, we are going to talk about effectivity assay for AAV vectors. So effectivity assay is to detect the infectious titer of AAV products. The infectious titer is calculated based on the CT value of serial diluted vector in Q-PCR. It is a critical quality attribute to AAV product release. The infectious titer represents the capability of the AAV product to infect the target cells. It represents the potency of the AAV product. The infectious titer is the important tool to determine the minimally effective dose of a clinical candidate. At the same time, AAV production requires cytopathogenic effects, which only occur after co-infection with a helper adenovirus or herpesvirus. Here is how we perform the effectivity assay. So prepare the AAV viral vector with a serial dilution, and then co-incubate the AAV vector with adenovirus and the cells for 48 hours. After the incubation, the cells will be lysed and the AAV vectors will be harvested. And then the supernatant will be seeded from Q-PCR or digital PCR. After the amplification, the effectivity will be calculated based on the CT value at different dilutions. After the amplification, the data analysis can be done for infectivity assay. Calculate the mean CT first, and then the standard deviation for each dilution, add only, and the negative control, which we usually use T buffer. The threshold CT is determined by subtracting 3 times standard deviation from add only mean. All wells having a CT value lower than the threshold value are scored as positive, and determine the positive ratio for all dilution levels from 6 to 12. After the amplification is done, we calculate the infectious titer of the test article by the formula here, and then calculate the specific infectivity of the test article by dividing the TA titer by the infectious titer, as shown here. Next, we are going to talk about the empty-full capsid ratio test. So during the production, we can see sometimes, except the full capsid, sometimes the empty capsid, partial different with different structure, and the capsid with the contaminant fragment can be generated during the production process. So therefore, the empty-full capsid characterization is an important parameter unique to AAV vector. So the empty and the partial capsid have significant effect on immunotoxicity, on transfection efficiency, on potency, and sometimes it will cause anti-AAV antibody generation and some other immune responses. That's why empty-full capsid characterization is important for AAV vector. Empty-full capsid ratio is detected by analytical ultracentrifugation, which is called ALC. But in recent years, some other new test methods have been developed to determine empty-full capsid ratio as well. The new test method developed include CIEF, CE-SDS, simple Western, and SEC mode. So the advantage of AUC method includes AUC can determine AAV capsid distribution, provides sufficient resolution to distinguish between empty, full, and partial capsids. AUC analysis is independent of the AAV serotype and the reagent spray. And the disadvantage of AUC includes that AUC demands a large sample volume, and the data interpretation requires expertise for AUC, and the test method is low throughput, and it is challenging to implement in GMP operations. Empty-full ratio can be determined by CIEF as well. CIEF represents capillary isoelectric focusing method. So CIEF is great to be used to separate empty, partial, and full capsid of AAV vector, provides rapid analysis, usually less than 1 number per sample. CIEF offers a good correlation of empty-full ratio of AAV, with orthogonal technologies. CIF requires much less volume of AAV vector, so that is a huge advantage for this method. The principle for CIF determining empty-full ratio is that the full capsids have a lower PI than the empty counterparts due to negative charges from the ssDNA they encapsulate. The empty-full ratio can be calculated by peak area by CIF method. And we can see from here, the ratio determined by CIF is comparable with the result from AOC and EX HPLC method. CIF can also be applied for other AAV testing, which include reproducible analysis of intact AAV and of denatured AAV. Murray's CIF method is also sensitive and linear. We can also use Murray's to monitor AAV particle stability, and also we can use Murray's monitor AAV lot-to-lot variation. The empty-full ratio can also be determined by simple Western. The simple Western requires only 3 microliters starting material of vector sample. It has its own advantage for empty-full ratio determination. The simple Western can detect as low as 7.2 E7 vector particles per ml, and the assay is fast, so it can detect 24 samples in less than 5 hours. The empty-full ratio by simple Western result is comparable with the result from other methods like AOC, CIF, and EX HPLC. Follow us about this method at frontagelab.com for more information. Next, we're going to give a brief introduction on CAR-T therapeutics. So the figure on the right shows the modification of the CAR-T genes on cytotoxic T cells. And then after amplification of the gene-modified T cell, the cells will be infused back to the patient on clinical. It shows the effect of CAR-T19 on CD19 expression cells in vivo, and it induces changes in systematic immune factors in patients. So the CAR-T19 cells retains anti-tumor activity in vivo. The study also characterized the relative subset of CAR-T19 cells. This slide shows the lenti packaging plasmid, which usually contains 3 or 4 plasmids. The transfer plasmid encodes the vector genome. The packaging plasmid sometimes includes 1 or 2 for packaging. And the other plasmid includes envelope plasmid, helper plasmid. The figure on the right shows the lentivirus vector. We can see the different segments of the plasmid. This slide shows the lentiviral vector production workflow and the RCL requirements at each step. So the workflow shows the plasmid development and production, and then the cell expansion, then the plasmid transfection, the viral vector production, after that the purification and the fill and the finish of the product. So the RCL assay, which represents the replication-competent lentivirus, is to detect any potential amplification of a retroviral contamination that may be below the level of detection in the retroviral vector supernatant. The assay is to minimum the safety concerns of retroviral replication in CAR-T production and clinical patients. So usually RCL is required by FDA to be detected at master cell bank, working cell bank, and during the vector production, and after the viral transduced cells. So sometimes it is also required by FDA that even after the patient was injected or infused with the CAR-T cells, the RCL has to be detected in the patient sample. This slide shows the construct of the lentivirus packaging plasmid. It shows it contains the VSVG and SV40. So that's how we determine the VSVG and SV40 by Q-PCR for RCL assay. On the right, it shows the brief testing process for RCL assay. So basically, we co-transfection with the plasmid and then harvest the cells after centrifuge aliquot, and then the assay will be performed by Q-PCR. Frontage can provide the analytical testing for lenti gene therapy product, which is shown in the test method here. So it includes the plasmid integrity assay, the titer by genomic by Q-PCR, and the B24 by ELISA, the purity assay by SDS page and CE-SDS, and the expression and function test by Western blot, ELISA, or bioactivity assays. It also includes the genomic integration assay by Southern blot or PCR, residue plasmid DNA by Q-PCR, cell genomic DNA by Q-PCR. Sterility assay include bacteria, fungi, identification, BioBurden, sterility, gram staining, Mycoplasma by Q-PCR, and Endotoxin by chromogenic LAIO assay. The purity assay also include the whole cell protein, residue Benzonase, cytotoxicity assay by T-cell apoptosis assay. Like mentioned, the replication competent lentivirus, RCL by Q-PCR, and the long-term stability program, which includes the infectious ability test and the color, appearance, pH, osmolality at different time points. As mentioned, the RCL is very important for lentiviral vector release and stability program. So the test started with the vector transduced into the cells, and after 7 days, 14 days, 21 days of amplification of the cells, the supernatant will be collected for Q-PCR on VSVG expression or for P24 expression by ELISA. At the same time, the 21-day supernatant can be transduced into the indicator cells after 7 days co-culture. The VSVG and P24 will be detected again. In Frontage, we are capable of performing the RCR testing abiding by the FDA guidance. So firstly, a minimum of 5 passages of permissive cell line, at least 5% of the cell supernatant of 3 ml needs to be tested. At least 1% of the total cells or 10 to the 8 cells need to be evaluated for EOPC cells. Either cell cultivation test or supernatant amplification will be conducted with CE-8166 cells. VSVG Q-PCR endpoint can be performed upon request, and the test can be finished around 2 months. Here shows the RCR results. So the CT value of the positive control remains low. The CT value of the spike remains low after passage of P9. And the vector product shows much higher CT value all the time than the positive control and spike, which shows the vector is RCL-free. Here is the RCR result with different plotting. So the GFP positive cells, the HIV capsid and the viral copies of the positive control are high, and the GMP positive cells, the HIV capsid and the viral copies of the viral product are low. So it shows also the RCL is low, is at very low level at the viral product. These are the references related with the presentation today. Thank you.
Okay, great. Thank you so much, Jessica, for a really informative talk there. This is going to bring us into the Q&A portion of today's session. If you do have a question for Jessica and weren't able to get it in during the talk, now's the time you want to go ahead and do so using the box at the bottom of your screen there. So we'll get through as many as we can get to here. So first question, Jessica, besides the analytical methods discussed in the presentation, what are the other critical methods for AAV vector quality control release that you can share?
Yeah, as mentioned in the presentation, so titer by digital PCR is another critical method for AAV release. Nowadays, it's been well developed, so far.
Okay, great, thank you. Let's see here, moving on, this attendee would like to know, can the CE-SDS be used for AAV vector empty full ratio determination?
Yes. So similar to CIEF, the CE-SDS can be also used for empty full ratio for AAV vector testing.
Okay, great, thank you. Let's see here, moving on. Except AAV and lentiviruses, are there other viruses that can be used to deliver target gene for gene therapy?
Yes, other than AAV and lentivirus, adenovirus and retrovirus can also be used for gene therapy. So all the 4 different types of vectors have different gene expression advantage. and the expression time are different, and the target gene capacity are different too.
Okay, great, thank you. Moving on, we got time for a couple more here. This attendee would like to know how stable are the viral vectors if they're stored correctly in less than -80 Celsius?
Right, that's a good question. So if stored properly, the viral vectors are very stable. So we had a chance tested the potency assay for AAV vector after 5 years of manufacturing. The results are actually comparable with the results from time 0. So basically, they are very stable.
Okay, great, thank you. We're going to take one more question before ending the session today, and this attendee would like to know, do you know of any side effects so far for the viral vectors that have been used in the clinical environment?
Yes, the side effect for gene therapy includes the genotoxicity. So basically the genotoxicity can be missed behind the therapeutic advantage of gene therapy. A standardized test for determination and the quantification of the risk of genotoxicity for therapeutic vector should be developed, and is very important for the long-term development of gene therapy.
Okay, great. Thank you so much, Jessica, for all those answers.
Thank
and that's
you.
going to bring us
Thank
to.
You so much,
Yep. No, thank you for your time. We appreciate it. So that's going to bring us to the conclusion of today's session, everybody. If we didn't get to your question, Jessica does have your contact details and your question, and she will be able to follow up with you directly. Thanks again, once more, Jessica, for today's presentation. Thank you again to today's sponsor, Frontage Laboratories, and thank you to everyone in the audience for your attendance and participation today. This session will be archived for 1 year on our website, contractpharma.com, and you will be receiving an email from us shortly when the session is available for on-demand viewing. And if you are interested, Jessica's made her talk available for PDF download. You can find that in the Resources tab on your screen there. And that's it. Thank you very much for joining us, everybody, and have a great rest of your day. Take care. Thanks again, Jessica.
Thank you, Tim.